Hello everybody, and welcome to this co-webinar with IDT and Volta Labs. My name is Nick Downey. I'm NGS Collaborations Lead at IDT. It's my pleasure to take you through some of the experiments we've been doing with Volta Labs to make workflows for NGS more simple. As most of you probably know, IDT has long been an oligo supplier and NGS components producer. We pride ourselves on our customization, our proprietary methods of synthesis, and our commitment to collaboration. We bring in high quality standards and vertical manufacturing with deep customer support to help our customers. We also have one of the broadest portfolios of NGS components on the market, everything from library prep to target enrichment. Our library preps span both DNA and RNA, as well as methylation. We have probably the largest offering of adapters and primers, supplying both stocked and custom solutions. We have a unique technology around normalizing library yield that we call Normalase. And in the target enrichment area, we have leading products both in hybridization as well as amplicon sequencing. If we look at our DNA library prep, we have a variety of approaches which can be used for whatever you need. Today we'll be looking primarily at our DNA library prep EZ kit, which is a rapid enzymatic fragmentation-based kit, and also looking at our cfDNA and FFPE DNA library prep kit, which is a highly effective and efficient kit which will capture the most complexity in your library. The first kit used in this study is our DNA library prep EZ. This kit has a simple workflow and takes genomic DNA into an enzymatic prep step, which both fragments and repairs and A-tails in one go. From here, we can do a regular ligation of either a stubby or a full-length adapter to create nascent library molecules, and then follow that with PCR to both amplify and/or index as needed. This kit is able to handle a broad range of inputs from 1 to 250 nanograms and ensures an even AT-GC coverage. A feature also is that the C of A will provide guidance on fragmentation time for each lot, which reduces the amount of time you have to spend trying to titrate the new lot to get the insert length you need. Here is some quick summary data of an experiment we did to look at the efficiency of library conversion using this kit. We took 1 nanogram of genomic DNA input and made libraries with both our kit and a competitor's. We then did a capture with our pan-cancer panel and sequenced to a depth of mean coverage of 45x. In this data, you can see clearly that IDT library prep allowed us to get a more complex library, resulting in deeper coverage. The other library prep used in this study was our cfDNA and FFPE DNA library prep kit. This kit utilizes already fragmented input DNA, which is then end-repaired to create blunt ends. We use proprietary, highly modified adapters and mutant ligase to drive the first step of ligation, adding on one of our adapters. This process allows us to have a higher level of adapter in the reaction and really drive the efficiency of library conversion whilst avoiding side reactions such as adapter dimers. Note we also have inline UMIs on these adapters allowing for error correction downstream if needed. In the second ligation step, we anneal the second adapter and copy across the UMI sequence before ligating to the insert, creating a complete molecule. We then use index primers to complete the preparation of this library. This data shows the increased efficiency of this library prep. Here we used 10 nanograms of cfDNA input into library prep and captured with a 61 kb custom panel. As you can see, when we calculate the number of unique molecules and thus the conversion rate of library prep, we see a much higher efficiency with our kit versus several of our competitors. On the right, you can see that we are able to achieve much deeper coverage with these libraries. So from here, I'm going to switch from our library prep to our hybridization enrichment. We have a variety of solutions in this space. The basic workflow is probably well known, but in brief, we take the libraries and mix in some blocking oligos which prevent adapters from forming daisy chains. We also add some COT DNA which will prevent repeats from daisy chaining as well. Then we add our hybridization probes and incubate to allow those to bind. Those probes are labeled with biotin, which we can then bind to beads via streptavidin. These bound fragments can then be amplified and sequenced specifically. Here's some example data from our exome hide panel V2, where we can see compared to various competitors, our hybridization efficiency and probe quality allows us to get deeper coverage across more of our target space. It's now my pleasure to hand the reins over to Abdul Mohammed of Volta Labs. Abdul serves as Head of Applications Development at Volta Labs, where he leads all efforts for application spin-up and testing. He joined Volta as a senior scientist in 2020. Previously, he was a senior scientist at UltraView, leading the development of UltraView's in situplex technology for deep profiling of tumor samples. Abdul holds a VTech and MTech in biotechnology from the Indian Institute of Technology. He also received a PhD in chemical and biomedical engineering from Johns Hopkins University, where he developed self-assembling DNA circuit interconnects with applications in nanofabrication and self-organizing biological components. Over to you, Abdul.
Thanks, Nick. This is Abdul from Volta Labs. It's been a pleasure collaborating with IDT. As you've shown, IDT has quite an impressive and wide product portfolio with the XGen product line, ranging from many different types of library prep, normalization, and hype capture. In my section of the webinar, we are going to focus on the IDT XGen hype capture workflow on Callisto and showcase some of its performance results. First, let me tell you a little bit about Volta Labs. Volta Labs is a Boston-based genomics applications company that simplifies sample preparation for sequencing. Our mission is to be the universal front-end platform for every sequencing technology. We commercially launched our platform Callisto in 2024. To date, we have enabled 4 applications, including high molecular weight DNA extractions, DNA library prep for Illumina, PacBio SmartBell 3.0 library prep, as well as IDT XGen NGS hype capture, which we'll talk about today. Note our application menu is continuously growing, and in 2025, we intend to expand into RNA-seq, 10x library prep, Oxford Nanopore library prep, and many others. To date, we've processed over 25,000 samples, across a wide range of applications. This has enabled us to optimize our applications with high robustness, so they are push button ready. Let me introduce you to the Callisto sample prep system. The product has three components. First is the Callisto platform. This is the benchtop instrument. This has a small footprint that easily fits on a standard lab bench. Second, the instrument comes with preloaded apps. We refer to workflows on Callisto as apps due to the push button nature. There is no need for method development or optimization. Third, kits. We provide kits for all of our applications. Kits contain plastics, consumables, and reagents needed to process samples on Callisto. Let me highlight a few advantages with the Callisto system. First, Callisto is a benchtop instrument with a small footprint that easily fits on a standard single lab bench. It's smaller than most liquid handlers, and the only requirement is to have an outlet. You don't need an integration engineer or an automation engineer or special training required for operation. Callisto comes loaded with pre-optimized apps. You don't need to do method development, unlike other traditional liquid handlers. No optimization or method development required. It's push and go, and our customers get started running applications on day one. Callisto supports multiple applications right off the box, from extractions to library prep to hype capture, all on the same platform. We've engineered Callisto such that we enable flexible batching up to 24 samples, so you don't need to wait to gather enough samples to process on Callisto. You can operate Callisto with 4 samples and as high as 24 samples in multiples of 4, with no impact on cost per reaction. Let's dive into the hype capture application on Callisto. Callisto's core technology combined digital fluidics, magnetic bead operations, and temperature control, all on a single reaction zone. This enables mixing, magnetic bead base separation, and consistent reaction temperatures throughout the entire workflow. On the left, you can see an overview of the target capture workflow by IDT. You start with unenriched libraries prepared using the appropriate library prep kit, depending on whether it's genomic DNA, cfDNA, or FFPE. You then add your blocking oligos to prevent daisy chaining wire adapters. Biotinylated oligos are then added to hybridize to your region of interest. IDT has off-the-shelf panels as well as ability to customize panels as per your needs. Probe target hybrids are captured using streptavidin-coated magnetic beads and washed several times to enrich the library for the desired targets. Traditionally, these wash steps meant that you have to remove the tubes from heating, vortex, pellet, and remove your supernatant. Not on Callisto. It's a closed system with a complete walkaway experience. We've automated this workflow on the left on Callisto and designed it such that operators can get started immediately. There is minimal training required, and run setup takes less than 30 minutes. This reduces the training requirements of running this workflow and saves hands-on time that can be spent elsewhere. Along with ease of setup, Callisto enables high-quality target enrichment that is independent of operator. For labs that do hybrid capture without automation, there's several challenges. First, the hybrid capture workflow is very temperature sensitive, has many temperature sensitive steps. Small deviations in your hype temperature or conditions can lead to suboptimal probe target interactions, affecting your downstream sequencing analysis. The workflow also has many complex steps, increasing the chances of user errors and having the need for high skill labor in the lab. Here's an overview of the IDT XGen Hype Capture app on Callisto. The input to the app are DNA libraries. During setup, the user chooses the duration of the hybridization, whether it's 4 hours or all the way up to 22 hours. The user then walks away for the duration of the hybridization. After hybridization is complete, Callisto prompts the user to load the capture reagents. Once the reagents are loaded, the user then walks away and Callisto processes the samples throughout the remaining part of the workflow, from capture to washes to the PCR setup. The output libraries are ready to be directly loaded onto the thermocycler. Let's dive into the data. To evaluate IDT XGen Hype Capture app performance on Callisto, we performed 3 studies. Phase 1 and 2 evaluate inner user variability of singleplex libraries of the Exome V2 Hype panel and the Pan Cancer Hype panel respectively. We compared directly to manually prepared libraries. In Phase 3, we evaluated multiplexing capabilities on Callisto from singleplex all the way up to 20plex libraries. Overall, the goal of these studies is to demonstrate how Callisto enables full walkaway automation while retaining the high performance of these IDT kits. Let's dive into the phase 1 data, which looks at the Exome V2 panel on Callisto. In this study, we use 100 nanograms of HG002 DNA prepared using the XGen DNA Library Prep Easy Kit as inputs, and then use the XGen Exome Hype Panel V2 for the target enrichment. This panel has approximately 415,000 individual probes spanning 34 megabases of target region for the human exome. Libraries were sequenced on the NextSeq 2000 with 2x150 cycles using a P4 kit. We then downsampled to 50 million paired end reads across all samples for comparison. Here we are looking at the primary results, including post-capture yield of libraries, as well as the mean target coverage after sequencing. Callisto prepared exomes yields are in pink, and manual data is shown in gray. The key takeaway is that Callisto provides consistent post-capture yields. This results in consistent sequence coverage across all samples. This data is a good indication that Callisto's hybridization app workflow reduces inner operator variability. Here we're looking at some of the key metrics that impact biology. Fold enrichment, that is how enriched the targets are relative to the genomic background, is highly consistent for all samples prepared on Callisto. This translates into higher consistency between capture samples. The consistent temperature throughout the full workflow enabled by Callisto delivers high quality captures and eliminates inner operator variability. Of particular interest is how even the coverage is across all the target regions, shown here with the fold 80 base penalty. Lower score is better. And this data shows that across users, Callisto shows comparable performance as manually prepared samples. The duplication level is also equivalent to manual. While 8% may seem high for some users, this is sequenced on the newer X-Leap chemistry, which has been shown to have higher duplicates than previous generations recently. The key here is that Callisto is performing just as good as manual across all replicates and users tested. Here we show Callisto produces enriched libraries that generate exceptional on-target reads for the exome panel. 94% of the bases sequenced align to a target region, which is exceptional enrichment over baseline whole genome sequencing. Almost all of the target bases were covered when downsampled to 20x, demonstrating how consistent and even the coverage is over all targets in the exome panel. Let's zoom into some target genes captured in this panel. Here we are looking at a single sample of Callisto versus manual. RB1 is a gene found to be mutated in type 2 diabetes family and thus is thought to be susceptibility gene for type 2 diabetes. This is traditionally a difficult to capture region because different parts of the exon have drastically different GC content. RB1 exon 1 has GC content near 77.5%, while RB1 exon 2 has GC near 35%. You can see in this comparison to manual a near identical performance and the ability to capture difficult to sequence regions. Callisto enables you to obtain high performance target enrichment libraries as expected with the XGen chemistry. In another example, we look at the TERT gene and promoter. Mutations in the TERT promoter are found in over 50 types of cancer. Again, we have a very high GC target, with the TERT promoter ranging from 71 to 83% GC content, and the exons hovering around 78% GC. Across the full region, you can see that there is sufficient sequencing coverage to identify mutations. This can be observed in the mutated bases found in the genomic pileup. Callisto enables you to access difficult to capture targets that are critical for understanding tumorigenesis. That concludes the phase 1 study. The key metrics for exome V2 panel are great. This demonstrates Callisto's performance with large panels, but what about smaller panels? That's what we're going to take a look at it in the phase 2 of this study. It was the same experimental design. However, we used the XGen pan-cancer hype panel. For inputs, we used reference DNA with 20 nanograms of Horizon's OncoSpan cfDNA, prepared using the cfDNA and FFPE DNA library prep kit. This panel has approximately 7,800 probes spanning 800 kilobases of target regions for the oncogenes. This was sequenced on the NextSeq 2000, 2x100 cycles using P1 kits. We downsample all to 4 million paired end reads across all the samples. For the primary metrics, you can see again that Callisto is producing just as much libraries as you expect relative to manual. After downsampling to 5 million reads per sample, the target coverage is very consistent between samples. Next, let's take a look at key metrics that impact biology. First, target enrichment, that is how enriched are the targets relative to the genomic background. With Callisto, you can see a dramatically improvement in consistency of your fold enrichment between samples with the pan-cancer panel. With regards to coverage evenness, that is fold 80 base penalty, Callisto performs equivalent to manual hybridization. The duplication level is also equivalent to manual. The key takeaway here is that Callisto is performing just as good as manual across all replicates and users tested. Callisto produces enriched libraries that generate exceptional on-target reads for the pan-cancer panel. For a smaller panel, like pan cancer, up to 90% of the bases sequenced aligned to the target region, which is exceptional enrichment over a baseline whole genome sequencing. Almost all of the target bases were covered when downsampled to 100x, demonstrating how consistent and even coverage is over all targets in a small panel. At 100x, you can detect those low frequency variants. Callisto performs equivalently to manual hybridization workflow across all coverage depths. Let's zoom into some target genes captured in this panel. MTOR regulates cell growth, proliferation, motility, survival, and protein synthesis, and much more. Note, with downsampling to the same coverage, there was sufficient coverage to call a mutation on Callisto prepared enriched libraries to identify low frequency variant alleles that are expected. The input DNA horizon has been validated, and the mutation expected here is a C to T at 15.5%. That's exactly what we find with Callisto to the dot. EGFR gene plays a critical role in cell growth and development, as well as its association with various cancer when mutated or overactive. The expected mutation frequency is 1%, which matches the experimental results obtained on Callisto. With Callisto, you can find low frequency variants for clinically relevant mutations. This concludes the phase 1 and phase 2 of our study that focused on measuring the inner operator variability of the hype capture workflow on Callisto. We've shown a high consistency between runs and with low run-to-run variability. We've seen this performance boost, all with saving 2.5 hours of hands-on time. We've also demonstrated this with large panels and smaller panels like cancer. Let's move to the phase 3 part of the study. A key aspect of the hype capture workflow is the ability to pool multiple libraries to save on capture costs. This is called multiplexing libraries, and it allows you to pool several barcoded libraries and capture with a single set of probes. As you increase the plexity of the pool, the ratio of probes to library decreases. It's therefore essential to make sure that the on-target rate stays high for all plexes. With a 20-plex, some of the samples are extremely viscous, with 10 micrograms of DNA going into the pool. In this study, we wanted to evaluate the performance of Callisto with the IDT hype capture app across one single plex to all the way to 20 plex. We used the Exome V2 panel for this study. Diving into the performance data. When looking at the on-target rates, one key takeaway is that Callisto delivers high on-target rates even at very high plexity libraries, all the way up to 20x. The key metric, sequencing evenness, remains very good, with fold 80 penalties in the optimal range and equivalent to or less than a manual singleplex. Manual singleplex is the best you can get, and as you can see, a 20x plexed library on Callisto performs as good as a manual singleplex. This concludes the phase 3 part of our study. One of the key takeaways from this study was that Callisto passes cost savings to our users by enabling high quality and high plexing of library pools per hype reaction. I wanted to highlight the key benefits of the IDT XGen Hype Capture app on Callisto. First, we've enabled a complete walkaway solution for hybrid capture. Hybrid capture is considered one of the most complex temperature-sensitive workflows among the NGS sample preps. Second, we've shown high consistency between runs and operators. Third, we've demonstrated uniformity of coverage uh, with the enriched libraries and capability to detect low frequency alleles across different panel sizes. Last, users can process from as low as 4 samples to 24 hybrid reactions per run on Callisto, allowing for flexible throughput to match your lab needs. As a recap, the implementation of the IDT Hype Capture app on Callisto starts with DNA libraries. This is followed by hybridization, capture, heated washes, room temperature washes, and PCR setup, all while enabling walkaway processing on Callisto. The output for the app are libraries ready for PCR amplification. As I mentioned in the beginning, we offer kits that go along with apps on Callisto. In case of IDT Action Hype Capture, we have designed a 16 reaction kit that includes all the plastic consumables you would need for 16 hybrid reactions on Callisto. With that, I want to thank the IDT team as well as the team at Volta Labs for their hard work in bringing this product to market. We're excited to see how customers scale and simplify hype capture workflows in their labs using the solution. To learn more, please reach out to Volta Labs, and I want to thank you for your time.
Abdul, thanks very much for sharing that data. This is a pretty exciting project. While I'm just waiting for a couple of questions to come in here, could you just spend a minute and maybe highlight the performance metrics from this app that you're most excited about?
Sure. Yeah, that was a really fun project for us to work on, Nick, and it's been a pleasure collaborating with you and the IDT team. So you asked the question, what metrics, I'm excited about. Maybe what I'll say is like what jumped out to me was the chemistry, the performance of the IDT action chemistry, both especially at the difficult to capture regions, and how well it did at the high GC areas as well as low GC areas. I was quite impressed, with the performance there. And, I'm glad we are sort of able to collaborate together and bring this capabilities of this chemistry through the Volta Callisto system to, and scale with automation. Nick, so question for you on similar lines, from your perspective, where do you think we will have the greatest impact? This will have the greatest impact for your customers.
Yeah, well, so I think in general, hype capture can be a little technically challenging with all of the temperature control, etc. And so I'm pretty excited to see the way that the Callisto allows you to be sort of hands-free, right? You load up the reaction and then you sort of walk away, and the machine handles all of those difficult steps. So I think it's going to really help those labs who may be a little newer to Hype or have newer lab people, and this will allow them to accelerate and get their data faster, which is always a good thing.
Cool.
All right. And here we go. So we've got some questions. I think this first one's for you, Abdul. Have you designed this process considering dead volume?
We get that question all the time. I think the answer is yes. What the impact of the customer, when you think about this, we've kept dead volumes in mind, and when a customer is using IDT's Xgen Hype Capture Kit with Callisto, you will be able to extract, if you're using a 16 reaction kit, you will be able to do 16 reactions from that kit. If you're using a 96 reaction kit, you will be able to use 96 reaction kit, even under the lowest batch size that we support. So we've designed the app thinking about this and keeping this in mind.
Yeah, I think that really helps. I think a lot of people have that kind of anxiety when they're
thinking about automation, because often that is a problem. So that's cool. Okay, I think this one might be for me. So the question is, how are you able to get 20 libraries per 16 hybrid reactions? And I think that this is also sometimes an area where people might misunderstand. And so just want to clarify, when we were talking about the 20 libraries, we were actually referring to 20 libraries in one capture. So we call that 20-plex captures. And so that's only one reaction. And so if you were doing that with a 16 hybrid reaction kit, you could actually get as many as 320 libraries captured through the system.
Yeah.
All right.
Maybe
So
maybe if I chime in there, it's that's like plexing really. We've seen our customers interested in plexing capabilities, and I'm glad IDT supports plexing. That's one way to bring down cost on hybrid reactions, and so that's great, Nick. We're excited about this too.
yeah, good call out. And so, another question. I think this one again is for you. How does Callisto support sustainability?
In sort of multiple ways, but maybe one thing that I want to call out is one of our unique capabilities using our technology is we actually dramatically reduce the number of tips used in a workflow compared to traditional liquid handlers, while having the benefit of the walk away and sort of improving capability and seeing improvement in performance. This is fundamental to the technology, and so sort of what the impact this has is you have fewer plastic used on a per reaction basis, and also you have to have less storage in your labs and less operationally less complex. So our customers seem to like that angle as well.
Yeah, I think more and more people are tracking that, and I think it's great that you thought of that as you set up your system. Another question, I think again is for you. Have you performed this capture with smaller panels?
Oh, good question. So as we saw in the study, we've tested in our development with exomes, which are about 400,000 probes on the larger side, and on the smaller side, we tested with the pan cancer panel, which has about 8,000 probes, and the performance remains robust throughout at the extreme at these two ranges. We expect this performance to continue to remain robust as the panel size goes smaller, but we've not tested very small panels as of yet.
Yeah, I think our customers like to stretch those limits, and so they're always looking for something else. But I agree. I'm excited about following up on that. The next question, which perhaps is maybe for me, the question is, can you capture RNA on the platform? And I think here, perhaps RNA is actually referring to RNA libraries, and those have obviously, just to be very clear, been converted to DNA at that point. And so in actual fact, the chemistry works equally well on an RNA-seq library versus a DNA-seq library. We haven't tested that yet on Callisto, but based on over decades worth of data, I think the XGen chemistry can easily support that. So that should be no problem. Okay. Oh, another question, and again, maybe for me this time. What if I have a different library prep that's not XGen? Oof, tough one for me to answer. Obviously, I want you to keep using IDT kits. But historically, before IDT built out its library prep portfolio, we did have capture, and we used third-party kits. For example, the now Roche Kappa library prep, and our chemistry worked very robustly for that. So I think we have good confidence that our capture system would work with different library preps. But of course, I would encourage you to take a look at what IDT can offer you. A lot of our kits are differentiated and could really help. Okay. This one actually is back to you, Abdul. Could you explain where the hybridization, the capture, and the wash happen on the instrument?
Cool. Great question. So from a hybridization, so when you look at Callisto, and you have two different sort of subsystems a user interacts with. One is, the 24 reaction zones all the way in the back of the system. That's where you do all your, magnetic bead-based operations, electric control beads with electric field, and so on and so forth. And then you have this deck in the front where you store your reagents and tips. But also there is a unique, thermal block that has capabilities of like cooling reagents as well as going up to 95C. So when you're setting up the IDT exchange chemistry on Callisto, you set up reactions before hybridization. So you put your hybrid reaction setup into the thermal block at room temperature, and then the thermal block will go. The hybridization occurs on the thermal block. It'll go through the 95C and 65C incubation, and then the capture and wash happen on the reaction zones. So the hybridization reactions are transferred all the way to the reaction zones, and then at the end of your washes, the sample is brought back onto the thermal block into PCR strip tubes, where you have your PCR master mix prepped, so user can collect it back from the thermal block. So when from a user workflow perspective, you're primarily interfacing from samples in at the thermal block and samples out at the thermal block for this particular application.
Cool. So yeah, that's pretty simple from a user perspective, and it's kind of cool to see it in real life. So look forward to that. Okay, next question, which is the perennial one whenever we work together with another group. The question is, who do I call when this doesn't work? So Abdul, do you want to take that one?
Sure. I think we have very strong collaboration with IDT, with our technical teams. But and so if you have run into technical issues, you can reach out at support@voltalabs.co. We can be the primary first response, and then we'll certainly pull in IDT technical team as and when necessary to promptly resolve issues.
That sounds great. Yeah, I think we're both sides are fully committed to supporting this and ensuring that customers have a good experience. Great. Well, thank you for that. I think that's the end of the questions, and I think we're out of time today. So I hope everybody enjoyed this webinar. Abdul, thank you so much for your time. Again, it's been a great collaboration. I'm super excited about the future working together. And I'll just say to everybody else, if you have more questions, feel free to reach out to either group. We'd be more than happy to help.
Great. Thanks, Nick.